All right. So why regulate protein synthesis? And how do eukaryotic cells like ours do that? Well, remember that you have, in every single one of the trillion cells that makes up your body, there are thousands, right? Maybe more than that, thousands of genes in your DNA. In the DNA of a cell. You could not possibly have a cell be using all of its genes to make all of its proteins all of the time, right? You certainly wouldn't do that. Cells only use genes to make those specific proteins, right? When those proteins are needed. And that depends on environmental stimuli, on internal homeostasis stimuli, on whatever the needs of the cell are, right? Otherwise, I mean, it's ridiculous for a cell to be using all of its genes all the time. It only needs some proteins. A pancreatic cell is going to be using the gene for insulin a lot because it's going to make a lot of insulin protein. A cell that's in the brain might be using a gene that manufactures neurotransmitter molecules. So it will be using that gene a lot, right? Skin cells are going to use very different proteins, different genes or different part of the human genome than a neuron or a pancreatic cell would use. So it's a way of responding to the environment. It's a way of conserving energy, and it's a way of not wasting energy. Okay, so now if we look at this diagram, it might seem overwhelming, right? Look at all this. Oh my gosh, what is going on here? So let's just make sure that we go back to the parts of it that we already did that you do know. So look, oops. There's transcription. We learned that, right? There's the first step of protein synthesis. There it is. The first step of protein synthesis, where you take the DNA and you make a messenger RNA from it, right? So you're going from DNA to messenger RNA, right? That's transcription. There is the first step of protein synthesis. You know that. We talked about that. So let's just make sure we know that that's there. We have then some other things that happen that we'll talk about in a minute, but then other than that, if you look a little bit further down, you will see that the RNA will leave and go into the cytoplasm here, right? And there is the second step of protein synthesis, which we have learned about, translation, right? Where that messenger RNA is translated into a polypeptide or a string of amino acids. You remember that? A string of amino acids. That is all a polypeptide is. Those little beads there, okay? And that happens at the ribosome. Remember the ribosome, that sort of barbapapa looking thing where the messenger RNA goes in there and the beads come out the top like that animation showed us. So there's the second part of protein synthesis, right? Protein synthesis, step 2, right there, where the actual polypeptide is made, okay? And out here in the cytoplasm, and the diagram of the cell is all around all of these steps. So here you've got the blue part here in the center is the nucleus, and here's the nuclear membrane, right? So we're inside the nucleus there, and that's where your DNA is located, and that's where transcription happens, where a transcript of the particular gene is made into messenger RNA, and then the RNA leaves the nucleus, goes out to the cytoplasm, and that's where translation happens, right? Well, so far so good. These 2 steps, we know these steps. Now we're going to add to these, okay? All right, so there's a couple levels of control here. Make some color here. So there are lots of points along the way, along the way of a eukaryotic central dogma, right? You should know by now what the central dogma is. Central dogma just means from the DNA to the RNA to the protein. That's all it is, that linear flow of information, where lots of points where the process can be stopped, or altered. Lots of points, right? First point, right here. You have to have the right chemical signal hitting the cell. Right here. You have to have the chromatin, which is tightly wrapped histones, be modified so that the DNA is not tightly wrapped around histones and the RNA polymerase can't even get to it, right? So there's another point of control before you even open the DNA. There's also, the, in terms of the transcription happening, transcription can't happen without something called transcription factors. Transcription factors. To even get the RNA poly to get going, it needs those, right? Even after, and then after transcription happens and you've got your messenger RNA, it turns out that you've got RNA editing. The messenger RNA can be edited. Things can be spliced out and spliced into it. That's something a bacteria can't do, but our cells can't. So you can even alter the final protein that gets made. Even down here, now all of this stuff here would be, everything here would be post-transcriptional, right? Regulation. That would be post-transcriptional regulation. Oops, where's my arrow? I can never find my mouse on this thing, okay? Then even after something is translated, it turns out that you can even have post-translational things happen. Post-translational things can happen. Right. Okay. So all along the way, you can even have a polypeptide. The final, the polypeptide and the protein can be altered even after the polypeptide is created. Some cells will add a hemigroup to something, or some cells will not. It just depends on what's there in the cell, how it gets folded, all those different kinds of things can happen. So lots of points along the way where you can make little changes, where you can stop the whole process, or you can even change the final proteins that get made. All right, let me go back to the top for a minute. Remember hormones? Remember signal transduction pathways? Here it is again. So let's say that you have a signal, which is very often a hormone, signals the cell to start making a protein, right? Making a protein. Now, this could be lots of different things. Example, testosterone is actually a hormone. Testosterone is a hormone that actually will go right through the cell membrane. You know that, you remember that, right? And it will go and actually testosterone will go straight to the DNA. Oops. Testosterone will meet up and activate a transcription factor, which is a different protein, activates it, and then goes and binds onto the DNA just upstream of the gene, which allows the RNA polymerase, which you know is doing the transcription work, RNA polymerase, can then bind and do its job. So testosterone is one of those hormones that actually goes and gets a transcription factor, binds with it, these guys over here, and initiates transcription. And there aren't any eukaryotic genes that can operate unless there's a transcription factor bound upstream to help the RNA polymerase then attach and start doing its job. So eukaryotes need those transcription factors, and often outside signals help get those transcription factors attached to that area of the DNA that's where the RNA poly can then bind. So there's one thing, you got to have some sort of signal to get it started. Now here, this thing in here, chromatin modification, is the next level of control that has to happen. Before you can even get the transcription factor onto the DNA, you have to make sure the DNA is uncoiled from its histones and that it's available. So genes can't be transcribed unless their DNA is uncoiled, right, from those histones, those histone proteins. You guys who are here, look up on the wall up there, see those gray balls? Right, so if the DNA that's curled tightly around those histones is like that, right, close to that picture, then it can't be transcribed because the histone's in the way, right? So you have to unpack it. Then once it's open, then you can start, get the transcription factors on there, and the RNA polymerase makes a initial primary transcript. Now we're here. So we've done transcription, that worked because the DNA was uncoiled from its histones and because the right signal came in and put a transcription factor where we needed it. The RNA polymerase did its thing, and now we have a primary transcript, Now if you look at this, I don't know if you can see this, but if you look along here, you see that some of the transcript is sort of a dark red color like that. And then some of it is this sort of pale pink color. There's these pale areas in the messenger RNA transcript. Okay, so the next picture that I'll show you will be a little bit more clear about this. But what's going to happen is those pale places are called introns. And introns are what's called non-coding parts of genes. They are almost always spliced out of the RNA before the messenger RNA goes out to find a ribosome. So the primary transcript has a bunch of pieces in it here, these pale pink pieces that are going to be spliced out. Look at what happens. RNA processing, the RNA is edited, and now those pink parts aren't there anymore, and what you have are all the exons that have been stitched together. The introns are removed. These guys are removed, they're spliced out, and the exons are then stitched back together so that all that's left is the coding part. And then there's some other little things added. There's a cap on one side added and a tail on the other, and we'll talk about that in the bigger picture. So now you have a final messenger RNA transcript. It's going to travel out to the nucleus, travel out to the cytoplasm, transport it out to the cytoplasm. Here it is again. Messenger RNA in the cytoplasm. It only has a short lifetime, so that's another point of regulation too, is that messenger RNA doesn't last very long in the cytoplasm. There's some digestive enzymes out there that are attracted to it, and they're going to break it up, And that's actually a good thing because you don't want to make a protein endlessly. You want to have an off switch. Well, in lots of places in biology, if you have an on switch, you have an off switch. So there's a natural degradation of messenger RNA. It doesn't last forever. But while it lasts, it's going to find a ribosome, just like over here, right? There's the ribosome, and there's the messenger RNA transcript is right there, and here comes the polypeptide that's being made, right? So there's translation. All right. So then you get a final polypeptide, and then even the polypeptide can be edited differently, can be folded differently. Can be made and can have different little chemicals added to it, depending on the cell that you're in and depending on the form of the protein that you need. Right? So again, there's eukaryotic gene expression, right? So you have protein synthesis, the two steps that we talked about, transcription and translation, but then you have lots of stuff before that and between that and at the end of that that can either stop the process altogether or kind of alter the final product, right? Okay, next page. Okay, down here we have this diagram that I guarantee you'll see on the exams. You'll see it on the AP exam, you'll see it on my exam. This is a very common picture. And the short ed puzzles that I put up for you today, thank you. The ed puzzles that I put up for you today, Professor Dave explains this picture really nicely, and so does another AP Bio teacher that I actually thought did a really good job with it. But what you have here is you still have a DNA strand here, okay? But we've taken away the nuclear membrane, taken away the pictures of the cell in the background. So we're just looking at molecules here. This DNA kind of is represented by these orange and pink rectangles here, but it's still the double helix. That's all we're looking at here is a section of DNA. So the genes, right, that we're about to encode, this right here is the gene that needs to be transcribed, right? Here it is. Gene to be transcribed is sitting there. And there are some pieces of it, just like in the picture above, that are exons. This is an exon. This is an intron that will eventually be spliced out. Here's another intron that will be spliced out. Here's an exon. There's 3 exons here. There we go. Okay, now if you look upstream from eukaryotic genes, right? There is something called a promoter region, okay? So upstream from the gene, this promoter is where RNA polymerase binds. Okay, it's just a section of DNA, that's all it is, right? RNA polymerase is going to bind there, but it can't bind there unless what? What also has to be there for the promoter to work? What has to attach to it before this guy can then attach to it and do its job? Yes, exactly. Very good. Transcription factors. And in many cases, there's more than one. Transcription factors, which are proteins, which are coming, sometimes they're coming from outside of the cell as signals. Sometimes they're existing in the cytoplasm or the nucleoplasm and just are waiting to be activated by other proteins that are coming from the outside, right? So those transcription factors kind of attach themselves to the promoter, and then the promoter becomes the right shape for this very important guy to come along, our RNA polymerase molecule, to start transcribing this pre-messenger RNA transcript, right? So here's DNA, there's the DNA, and there's the gene along the DNA that's transcribed, and now we are going through transcription, which needed some help from transcription factors before it could happen, right? Okay, so now you've got that preliminary messenger RNA transcript, right? Use a different color. So here's your messenger RNA pre, meaning it's not finished. Not finished. It has to be edited. So we're still in the nucleus here. So now we're going to be, they call it RNA processing. We're going to say it's called editing, right? So the introns are going to be removed and the exons spliced together so that you get the final messenger RNA transcript. There. Okay. A couple other things are put onto that transcript. This thing here is called a polyadenine tail. It's just a bunch of adenine nucleotides that are added onto the end. It's thought that what they do is they attract the digestive enzymes in the cytoplasm first, so that it kind of lengthens the lifespan of the messenger RNA. It allows it to do its job for a little longer than if the tail wasn't there. Then on this side, you also have something called a five-end cap. It's called a cap. And the cap, its thought is helps the messenger RNA to attach to the ribosome. Helps attach. Take this up a little bit. Almost done with this. Helps to attach to the ribosome. All right. Okay, so once you have a finished transcript, it leaves and goes into the cytoplasm. It finds a ribosome, and then translation happens, and now you have the final polypeptide, which is like a string of beads, a string of amino acids that then needs to get folded, properly folded and modified before it will work. Because you know that no protein can work unless it has the right three-dimensional structure and has the right functional groups added to it, right? Okay, so there you go. That is the basics of eukaryotic gene expression regulation. So we've gone a bit beyond just simple transcription and translation. You can see that betwixt and between there's quite a lot of those steps that can happen. So are there any preliminary questions right now, or is it, do you have a good idea of where we're starting? Want to give me a thumbs up or a thumbs sideways or a sort of a, yes, I think I get it? Okay, all right, good. All right, that's fine. Okay, that's a good question, actually. What are introns for? Yeah, what are they doing in the first place? If all we're going to do is splice them out. What a great question. Okay, so here's the thing, is what we think is that introns are not always completely spliced out. Sometimes in one cell, if they need the final protein to be a slightly different shape, they'll leave one of the introns in and then take the others out. So having introns at all gives a eukaryotic cell the opportunity to do different, to make different, slightly different proteins from the same gene, which is a good thing, an advantageous thing. Because as you know, the more diverse variation you have, even within the human body, the more adaptive you can be. Does that make sense? I believe that probably answers the question as well as I can. Right, normally it's non-coded DNA, but every once in a while you have cells that will differentially splice out introns and leave others in. And that would obviously change the shape of the protein, because as you know, the primary sequence of a protein is going to affect the final tertiary and quaternary shapes of the protein. Excellent question, wonderful. Okay. I think I'm going to stop recording now, unless anyone else has a really brilliant question, because that was so brilliant. But that's okay. I think that other questions might come up as you go into the rest of